Good morning, good afternoon, and good evening. Welcome back to Chapter 3. Let's see if we can't wrap this up. We are talking about proteins right now. We've just finished up talking about primary, secondary, and tertiary structure. I wanted to show that the tertiary is referred to as the functional conformation. The shape, conform, it has to be in to work. We said at the end of the last video that every single protein uses the first three. Amino acid sequence, beta pleated sheet, alpha helix, and then this functional conformation, the shape that has to be in to work. Some proteins additionally use a fourth level of structure, and this fourth level of structure is called quaternary. A quarter, 4 quarters, and a dollar, quaternary. This is where multiple protein chains interact to make one functional unit. And I've been using this idea, I've been using this concept as proteins are tools, right? A hammer is a tool, a wrench is a tool, a screwdriver is a tool. There are some tools that you have to put more than one piece together to make a functional unit. And a great example of that would be like a ratchet. You've got the handle of the ratchet that has a very special shape it has to be in to work. And then you've got the socket. The socket has a very special shape it has to be in to work. The handle by itself isn't terribly useful. The socket by itself is not terribly useful. The only way to make a functional tool is by putting the two separate pieces together. I put the socket on the handle. I've now made a functional tool. Not all tools have to be built like that. A saw is one piece. A hammer is one piece. A screwdriver is one piece. But some tools do. You got to put separate pieces together to make a functional unit. You got to put the drill bit in the drill to make a functional tool. The drill has a special shape it has to be in to work. The bit has a special shape it has to be in to work, but only by putting the two things together can I make a functional tool. Quaternary structure. More than one protein chain linked together to make a functional unit. A perfect example of this is hemoglobin. The protein that carries the oxygen in your blood. It's actually 4 separate protein units that have to bind together to make one functional unit. And if you make any one of them wrong, you can screw up the whole process. We move on from proteins, and now let's talk about our last biologically important molecule, and these are nucleic acids. Nucleic acids, there are 2 varieties of nucleic acids. There's DNA and RNA, and I'm sure you've heard of both of these. DNA stands for deoxyribonucleic acid. The deoxyribo refers to the sugar that's used to make the nucleic acid. The other variety, RNA, is ribonucleic acid. The ribo refers to the sugar. 2 different sugars, deoxyribose and ribose. I use 2 different sugars, and this is how I get my 2 different molecules. DNA is the master copy of the instructions that a living thing needs to do to stay alive. The DNA is the master copy. DNA is double-stranded. DNA is made of adenine, thymine, guanine, and cytosine. RNA is a copy of the master instructions. We'll talk about this a lot later on in the semester, but it's like working on a job site, on a work site, right? So there's a master set of blueprints that's held at the office. And when a workman needs a particular set of plans to work on a particular piece of the building, he doesn't go and take the master set of blueprints to the job site. What does he do with the master set of blueprints? He goes and he makes a copy of it. He puts it on the photocopier and takes that photocopy with him when he goes back to the job site. What happens if he takes the master copy to the job site and it gets damaged or destroyed? He's just out of luck. That's it. You're done. The job can't be finished now because the original instructions have been damaged. What happens if he takes a copy to the job site and it gets damaged or destroyed? No big deal. Just go back to the office and get another copy. RNA is the copy. DNA is the master set of instructions that living things use to make the products they need to stay alive. RNA is a copy of the instructions that living things use to stay alive. DNA is double-stranded, RNA is single-stranded. DNA is made of adenine, thymine, guanine, and cytosine. RNA is made of adenine, uracil, guanine, and cytosine. There's only one kind of DNA. DNA is DNA. There are at least 5 kinds of RNA, and we're going to talk about 3 kinds in our class. RNA, M stands for messenger. tRNA, T stands for transfer. rRNA, R stands for ribosomal, as in the DNA, the RNA that we find in ribosomes. And we'll talk about ribosomes another day. So this is showing how the flow of information goes. The original instructions is on a gene, and that original instructions is on DNA. Because I don't take the master copy to the job site, I have to make a copy of it that I can take to the job site. And that copy is RNA. I then follow the instructions on the copy to make my finished product, to make my protein. We just got done talking about proteins, and we'll talk about how we follow this chain of events, how we follow this flow of information in a later chapter, how we get from DNA to RNA to protein. This shows the backbone for DNA. This shows, I want you to imagine that DNA is a ladder where I've got two rails and I've got steps that I walk on. But because it's twisted, as we saw in this previous slide, because it's twisted, imagine it is a twisted rope ladder. I've taken a big tree, I've nailed the bottom rung of my ladder to the bottom of the tree, and now I'm going to wrap my ladder in a spiral as I go around this huge tree. That's how DNA is constructed. That's what this is a helix. It's called a double helix. It's a double helix because it's like a ladder. This is half of my ladder. This is one side of my ladder. This is the sugar phosphate backbone. The sugar phosphate backbone, the blue thing represents the sugar. In DNA, it's specifically the sugar is called deoxyribose. The nitrogen bases, the nitrogenous bases, are adenine, thymine, guanine, and cytosine. And as you notice, G can only bind to C, and A can only bind to T. And when I say A only binds to T, we're talking about on the other side. So if I were to build the other side of this, which is missing, it's really easy. What do I attach to this other side where this T is? Well, what's the only thing that can fit in there? A. What do I put on the other side of this G? Well, what's the only thing that can fit? C. What do I put the other side of C? G. Other side of T? A, T. If you have half of a strand of DNA, you can figure out the other side, because A always binds to T, A always binds to T, and C always binds to G on the other side. DNA is double-stranded. This is showing just one side. Here's both sides. Here is, again, imagine I have taken a rope ladder and I have nailed the bottom rung to the bottom of the tree, and now I've taken my rope ladder and I've wrapped it in a spiral around the trunk of my big old pine tree. All right? That's what I see here. A always binds to T, G always binds to C. This is, if you can imagine, insert the trunk of a tree into here, and that's how it's wrapped around. DNA is double-stranded. DNA uses thymine. RNA is single-stranded. This is half of my ladder. It's not the top half or the bottom half. It's the right half or the left half. DNA is double-stranded. RNA is single-stranded. The single strand of RNA, anywhere you would put a T, you replace it with U. You replace it with uracil. Anywhere you would put a thymine, instead you put a uracil. I still have adenine, I still have guanine, I still have cytosine, but instead of thymine, I put instead uracil. DNA is double-stranded, RNA is single-stranded. There's only one kind of DNA. DNA is DNA. It's all made the same way. Whether it's DNA in you or DNA in a bacterium, it's all made the same way. This is what DNA looks like. RNA is single-stranded. There are a few different kinds of RNAs depending on their job, depending on what they're supposed to do. Thanks a lot. See you in Chapter 4.